Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 27th January 2021. As we all know that we are into the revision mode, and we are revising the each chapter of the entire syllabus in the form of test papers. Already we have covered test paper 1, and in this session we are covering test paper 2. This gives a fair idea about how the question paper will be and what are the ways and means of writing the answers, presenting the answers, and it gives us an opportunity what are the topics that are to be focused in the given chapter. So we get familiarized with 1 mark questions, 2 mark questions and 3 mark questions. Also this gives an idea that which are the chapters in which 5 marks questions will come. So as you are seeing here, this chapter doesn't hold good for a long answer type, but it doesn't mean that, as I have been telling you, expect the unexpected. Sometimes you expect something, it comes, you are happy, but sometimes you don't expect something and it comes, we tend to be unhappy. So be prepared for the expect the unexpected also. So question 1, identify the polymer which makes exoskeleton of insects, exo, outer. Question 2, name the following. 1, sugar present in DNA. The name itself is there, expand deoxyribose. Second, base not found in DNA. So difference between RNA and DNA. Third, DNA containing the base pairs, 2 base pairs, okay. Then question 3, why proteins are called biological polymers. Question 4, which molecule has the ability to make their copies, own copies. Question 5th, what is metabolism. Catabolism plus anabolism. Mention the role of enzymes in metabolism. Mention the role of enzymes in, that is N, in metabolism. Name the 2 types of pathways occur in metabolism. Question 6th, why are enzymes called as biocatalysts. Question 7, give the functions of carbohydrates. Question 8, what do you mean by activation energy? What do you mean by, or what is meant? Okay, list the different types of lipids. Okay, or types of lipids. Question 10, describe the structure and function of ATP. And then most important one, differentiate between cofactor, coenzyme and prosthetic group. So take a paper, start writing the answers. You may take around 30 to 40 minutes. And after that, come back to this session. Keep your answers with you and compare the answers with the answers that I am giving here. Question 1, chitin, a polymer of glucosamine that forms exoskeleton of insects. Chitin, very tough exoskeleton. And question 2, 1 deoxyribose sugar. As we said, 1 oxygen atom is removed, so deoxy. Basically it is ribose sugar, okay, pentose. Second is uracil, the difference between DNA and RNA. And third one is aDNA, adenine. Question 3, as proteins are made up of multiple amino acids and they are able to perform multiple functions, that is protection, mechanical support, transportation, movement, they are called as biological polymers. Next, expansion of DNA, deoxyribonucleic acid. 5th, metabolism is defined as the sum total of living processes in the body. Enzymes trigger metabolic pathways. Enzymes act as catalysts. They are like cricket umpires, I told you. Cricket umpires will ensure that the cricket game goes on smoothly. Similarly, enzymes ensure that biochemical reactions go on smoothly in our body. Enzymes are highly specialized organic catalysts produced by living cells. The two types of pathways occur in metabolism are catabolism and anabolism. Catabolic pathway and anabolic pathway. Biochemical pathways refer to the reactions occurring in the cell in sequences. Enzymes guide the biochemical pathways along desired directions, as I said just now, and they have active site. The substrate binds to active site of enzyme and form enzyme substrate complex. Enzyme E, substrate S, C complex, which further changes into desired product. So here you must remember that catabolism, cat break. Catabolism is breaking, anabolism is making. So catabolic pathway is the bigger molecules such as proteins are broken down to amino acids. Fats are broken down to fatty acids. Carbohydrates, polymers are broken down to monomers. This is catabolism. Anabolism is small molecules joined together form a mega molecule. Like amino acids join together to form proteins. So together catabolism plus anabolism makes metabolism. In all the three, be it metabolism, be it catabolism, be it anabolism, in all the three, enzymes play a crucial role. Question 6th. Substances which change the rate of chemical reaction without altering the equilibrium point of a reaction is called catalysts. The catalysts of the organism are called enzymes. So we say biocatalysts. They are synthesized in the living cells, hence they are called biocatalysts. Question 7. Carbohydrates play important role in all metabolic reactions of body and formed as intermediate compounds in the pathways of the processes. 2. Ribose, deoxyribose sugars are found in DNA and RNA or RNA and DNA. Glucose is oxidized during respiration to yield energy. Already we said oxidation of one glucose molecule will fetch us 36 ATP molecules. So very, very important. Without glucose, we will not have energy. Glucose is used in synthesis of fats as well as proteins. Question 8. Activation energy is the energy required to initiate a chemical or a biochemical reaction. It overcomes the energy barriers of the reactants which occurs amongst the reactants due to presence of electrons over the surface. Second, absence of precise and forcible collisions, essential for bringing the reactive sites of the chemicals together. Next, most important one, lipids. Lipids are nothing but fats. So there are three types. Simple lipids, these are alcohols or triglycerides containing fatty acid and glycerol. Compound lipids, they are simple lipids with a biologically active compound in them. Example, glycolipids, that is carbohydrate lipid. Lipoprotein, proteins plus lipids. And third, derived lipids. They are hydrolyzed products of simple lipids such as fatty acids and alcohol. Question number 10. ATP, adenosine triphosphate, is primary and universal carrier of chemical energy in the cell, leaving cell capture, store and transport energy in a chemical form, largely ATP. It is the ATP which is the carrier and intermediate source of chemical energy to those reactions in the cell which do not occur simultaneously. These reactions can take place only if chemical energy is released. ATP molecule consists of a nitrogenous base, adenine, and a pentose sugar of ribose type, and of course 3 inorganic phosphate. That's why we say adenosine triphosphate. So it is adenine, pentose sugar, okay, ribose sugar, and 3 inorganic phosphate. That makes adenosine triphosphate. And 2 phosphate bonds are high energy bonds. One is relatively poor in energy. Energy released in living cell is thus stored in the chemical bonds of ATP molecule, which then serve as major energy yielding and energy requiring substance in the cell. ATP is broken down into ADP whenever energy is needed. ATP breaks down to ADP plus inorganic phosphate plus energy. So somewhere the energy will be in the form of calories, around say 4.1 calories roughly is released. That means the main purpose of ATP is energy release. That's why we call ATP as the energy currency of the cell. Question number 11. Difference between cofactor, coenzyme and prosthetic group. Cofactor, it is the non-protein substance. Cofactor is a non-protein substance that gets attached to an enzyme. Whereas coenzyme, as the name itself says, it's a protein group which is loosely attached to the open enzyme and a functional enzyme. Prosthetic, it's a non-protein part or group which gets attached to open enzyme. Second, it is essential. Cofactor is essential for functioning. It may be organic or inorganic or metallic factor. Coenzyme, NAD is a coenzyme for dehydrogenase, removal of hydrogen. Nicotinamide, adenine, dehydrogenase. And prosthetic group, some prosthetic groups have porphyrin of the cytochrome. The ring structures will be there. So this is how we differentiate the cofactor and coenzyme and prosthetic group. And in this chapter, the larger question that you can expect is the formation of substrate enzyme complex, lock and key mechanism of enzymatic action. So practice that. Plus type of proteins, structure of proteins, various types of proteins are there relative to structure. Similarly, fats, various types of fats and their function. These are